Hi everybody. In this video we're going to go over gene expression, which basically means how do you make the proteins that you're made of. And just to remind you of that, we are mostly protein. Now our cells do have around them membranes, and these membranes happen to be, in many cases, made of phospholipids and other materials. But the majority of our cells, other than the water, is protein and some RNAs. Even outside of our cells and between our cells and wrapping our cells, we tend to have a whole lot of proteins as well. So again, the majority of our cytoplasm is a gel made of proteins and some RNAs. And what gets a little bit complex here is to realize that the instructions for making those proteins aren't in the cytoplasm at all. They're found in the nucleus, and these instructions are written in DNA. And these long pieces of DNA are called chromosomes. And that's how we're going to start for today. So we're going to imagine yanking one of those chromosomes out of the nucleus and placing it right here. Keep in mind that this chromosome is a double strand of DNA, a double helix, of course, in terms of the structure, and that the two strands are in opposite directions. You can call that anti-parallel. So here's our DNA, and it is a giant, long, double polymer, or chain, of nucleotides. And that's going to be important a little bit later on. We'll talk about them in a bit. But before we get into that, remember that the chromosome is an incredibly long piece of DNA. On average, in your cells, we're talking about 130 million pairs of nucleotides for a single chromosome. That's average. Now, different regions of this chromosome are called genes, and these are the recipes for making those proteins that we're made of. So I'm going to make up some numbers, because per chromosome you might have about 1,000 genes or so. And so here are 3 genes. On this chromosome, you might have 1,000 of them, and each one of these is a recipe for making a protein. Okay. The recipes are in the nucleus, though, and the proteins are made or built out in the cytoplasm. So we're going to have some other form that we can use to sort of send the recipe. We're going to have to make a copy of the recipe. So that form is called specifically mRNA. Each mRNA is not a copy of an entire chromosome. It is not a massively long piece of RNA. It is a shorter stretch of RNA that matches a gene. That's it. Just one gene's worth of copying. And it is made of almost the same stuff. So eventually, that's going to be the material that we use that tells us how to make a protein, as seen here. And those proteins are going to be a little bit more complex to talk about because they're made up of a different material. But let's not get ahead of ourselves. Let's start with that first step. Going from DNA and making a copy of a gene sequence in the form of RNA. Now, DNA and RNA are both made of the same building blocks, nucleotides, and there are 4 different nucleotides for each, and what differs between the 4 nucleotides are these things called nitrogenous bases, of which there are adenine, guanine, cytosine, and thymine in DNA. You can just refer to them as A, G, C, and T, nice and simple. With regards to RNA, there are a few minor differences. mRNA specifically tends to be single-stranded, so you're not getting a double helix with the mRNA. Secondly, the backbone, not the stuff in the middle, not the nitrogenous bases, but the stuff on the outside, the backbone, has a different sugar in it called ribose. That's why it's called RNA as opposed to deoxyribose in DNA. These are minor issues, though. For what we're doing in this video, the only significant difference between the DNA and the RNA, other than single and double-strandedness, is that RNA happens to have a different version of a nucleotide. RNA happens to use U instead of T. Why that is nothing that you have to worry about for this class. The evolution of DNA from RNA and the multi-billion year history of that is well beyond the range of this class. Okay, so DNA, 4 different nucleotides. The differences between them are the A, G, Cs, and Ts within those nucleotides. And with RNA, pretty much the same deal, just a U instead of a T. So how do you actually do the making the RNA? So this process has a name, and it is called transcription. Transcription really means to just rewrite something. We're not changing languages or anything, we're just making a copy. So we're transcribing, rewriting a gene sequence. Now, the machines that do this are called RNA polymerases because you're making a new chain or polymer of RNA. We're not going to go into the details of all of that process, but I can sketch out the basics of it. So here's a gene sequence, and let's say the actual important recipe starts with an ACTTAG. The other strand, which is not the recipe itself, is in the opposite direction, CTAAGT. The strand on the left is the actual recipe or the coding strand. The strand on the right is just its match, also known as the template. Now, what's actually a little bit surprising is the actual recipe, the ACTTAG in this case, is not the strand that you read. You actually use the other strand. You pull apart the two strands temporarily, read the secondary strand, and then match up. And across from the T, you put an A. Across from the G, you put a C. Across from the A, remember this is RNA, you put a U there, etc., etc. If you look carefully or you rewind, you'll find that this new RNA strand in blue is nearly identical to the original recipe or coding strand that was previously there. So what we're doing is we're using the opposite strand as sort of a matching template to make the copy of the recipe. If that's too crazy to remember, just think of as making a copy of the recipe. That's the simple version. So next up, we're going to have to figure out a way of translating that into protein code. Now, the problem here is while RNAs and DNAs are just made up of 4 different kinds of nucleotides, proteins, on the other hand, are made up of a bunch of amino acids. They include glycine, alanine, valine, isoleucine, leucine, methionine, etc., etc., etc. There are 20 common ones that you do not need to memorize, but there are 20 of them, as opposed to just 4 nucleotide varieties in each of DNA and RNA. Now, the reason that's a problem is because we're going to have to translate between the 2 languages, and that's the actual term. We're going to do translation. The machines that do this happen to be called ribosomes and tRNAs. You should know that, but we're not going to go into the nitty-gritty details of the machinery themselves, how those machines work. Okay, why is this a difficult translation? Well, if you actually think about languages, there are certain translations that aren't so bad to do in terms of the alphabets. Going from Greek to Roman alphabet is not that bad because the Greek alphabet has 24 and the Roman alphabet has 23 letters, that is. In English, it's 26 letters. So if you're just transitioning one letter style to another, it's not a big deal. However, going from, say, English to my family's mother tongue, Telugu, is quite a bit different. You're not going from, beta to B or alpha to A. That's a 1-to-1 corresponding thing. That's pretty easy. In this case, we're going from 26 to 52, and that's a much more difficult thing to do. So for any Telugu letter like this, you might actually need 2 letters in English to represent the same sound or syllable or letter, however you want to put it. Okay, if that's too much to take, I'm just going to show you an RNA sequence, and here's a starting point. To actually encode each of the 20 amino acids, we're going to need 3 letters. We can't use 1, as I described in the language sort of analogy. 2 isn't even good enough because if you did 2-letter combos, that would only get you 16 combinations, AA, AT, AC, AG, etc. So we're going to do this in 3s. So I've broken this up into 3s, and we're going to translate each 3-letter code to mean 1 amino acid. Now, each of these 3-letter codes or words is called a codon. That's the vocab word for this. And each of those is going to be used by the translation machinery, the ribosomes and the tRNAs, to place the appropriate amino acid in place. So the AUG happens to encode something called methionine. The CAU encodes histidine, etc. You do not have to memorize this. You will have to learn how to use a genetic code chart, which is something that you can learn very quickly. Let's do one more example, and this next one's going to be real. I'm not making this up. This is a real partial gene sequence here. If we break it up into threes, we can see that I've got 4 codons here, 4 sort of sequences that tell us which amino acids go in this protein. This is not the complete gene sequence or the complete mRNA sequence at all. It's just a little bit. Now the ACU, when you translate it, is used to put a threonine into place. UCA, serine, etc. But here's the issue for today. I want you to imagine if you changed any of these letters. Now, if the gene itself in the DNA had the wrong letter at this U, honestly, it doesn't make a difference. But at this next U, it would. Now, when you look at the genetic code chart, you'll see why. But if we switched the U in the UGC codon to a C, that does not get translated into a C cysteine anymore. So that if you look on the right, the TSCG gets changed. Instead of a cysteine, you put in a much larger amino acid called arginine. Now, why should you care about any of this? This is all very lost in the weeds details of alphabets and craziness. This is a real example. If you happen to have a C instead of a U at that particular location on a chromosome, if you replace the T, which becomes a U, with a C, you get a wrong recipe. This is the recipe specifically for the green light receptor in the back of your eyes and your retinas. And if this is replaced, you get a protein that doesn't work as well. And the result of that, as you might be able to suspect at this point, is one of the most common versions of color blindness. Now, if you happen to be colorblind or have red-green color blindness, I don't know if this is the version that you have. It is one of the versions that are out there, but there are a lot of different sort of errors or mutations in this gene that can make this protein not work. And you can go through example after example of this. Overall, just remember that when you get the recipe wrong, or there are errors, or there are just differences, these cause our proteins to be different, which give us different characteristics. That might be a disorder or a disease. It might just be one of the things that make us who we are. Okay, big picture overview. In terms of the proteins, it all starts with the DNA. The order of the nucleotides in the DNA sequence of the gene, so the A, C, T's, and G's, determines the order of the amino acids. Which amino acids are you going to have in the protein? Most importantly, that determines the protein's shape or structure, and that's what tells you if it can do what it's supposed to do properly or if it does something a little bit differently. That's what's going to determine our characteristics in many ways, and that's how you get a protein. Hopefully that helped everybody. Feel free to rewind, rewatch what you need, or bring questions to class. See you next time.